The computer people's word. All right. All right, I just want to thank everybody for coming here today to hear about our research study in the roles of giant Starship, retrotransposons, that's my study, transposons and blasphemy and genome evolution from the sublime to the ridiculous. You see right there. All right, so I want to begin, button not work.
It'll work.
Button work now. Okay. I want to begin by introducing our focal species for this research study. It's Magnaportha oryzae. Magnaportha oryzae is a filamentous ascomycete fungus with a pathogenic life history. It is the etiological agent that causes blast disease in poaceae crops and their wild relatives. It is important to study this disease because it has severe economic and agricultural impacts, including like a study in, I think it's 2018, where wheat crops were found to have a 16% prevalence rate with 100% mortality of those infected. And then among rice cultivars, it was found that the yearly yield loss was around 10 to 30% just due to this specific fungi. So it's really a threat to global food security around the world. So moving forward to, now that we've gotten over some basics about Magnaportha oryzae, we will move forward towards its lifestyle to better understand the species. So Magnaportha oryzae is like many fungus, it can reproduce sexually or asexually, but it depends on the environmental and conditions and the availability of a mate. Magnaportha oryzae went through a bottleneck effect and historically, it went through a bottleneck effect and it lost its ability to produce parathecium outside of its origin of century. And it is also there's also another limitation that it's also a heterothallic fungi. So not only does two strains of Magnaportha oryzae need to be infecting the same host tissue, but one of the other strains needs to also be able to produce a parathecium. So this species is a mostly colonial organism, and it still exhibits a very high pathogenic variability and rapidly overcomes resistance genes in the cultivars being bred for blast resistance. So you may be asking yourself, without sexual recombination, how does this occur? Well, let's look at other classifications of pathogens to better understand how they may overcome this lack of sexual recombination. So classifications of pathogens by their host range, which would be either specialists or generalists. Specialists only have a host range of either one species or one cultivar of that species. And generalists typically have a wider host range, which can be in multiple families or in multiple species of a family. Now, Magnaportha oryzae is considered a specialist, but one thing about specialists is that they can be still possess the mechanisms by which generalists can use to host jump and host expand. So Magnaportha oryzae is a species that has many different strains that have host specialized populations. Therefore, the whole species has a very wide host range. So how do they overcome this? Well, one mechanism of pathogenicity, the use of transposons is used by pathogens to produce more variability in the genome. So Barbara McClintock discovered mobile genetic elements first in the early 1930s. That's when her research started. And then she discovered it in maize, and the color of corn being messed with due to transposons inserting into that gene. Since Barbara McClintock's initial study, we have learned more about transposons. Now we have two different classifications of transposons. Class 1 is an RNA transposon, which is characterized by its transposition intermediate, which is an RNA intermediate here. So it's more of a copy-paste transposon. It is copied into an RNA intermediate, then a DNA intermediate, and then it's inserted into its target DNA site. And then your DNA transposons are your class 2 transposons, which have a DNA intermediate. They cut themselves from their target DNA site, and when they want to move, they move as a whole. They don't copy themselves too frequently, move to a different target DNA site and insert themselves. So if we look at the transposons genes and their mechanisms of transposition, as I said, type 1 are copy paste and type 2 are cut and paste. LTR retrotransposons are characterized by the presence of long terminal repeats. So LTR retrotransposons are a subclass of retrotransposons, and non-LTR transposons do not have long terminal repeats on the ends of their genes, as you can see here. So there's many transposons throughout the genome of Magnaportha cerevisiae, and they are indicated by looking at the genes GAG and POL, or GAG, POL, and NB, or a reverse transcriptase gene. And then DNA transposons are characterized by their gene, the transposase, which is an enzyme used to transpose. So the relevance to our study for this stuff is that the Starship transposons discovered in the second paper here are a novel class of DNA transposons, or so we think. So the first paper here is the two, this one and this one are the ones that we use to build our study off of. So to get into the observations that led to our study, I'll move a little bit further. So the first paper had an observation that the avirulence genes were lost due to a transposon insertion, and then, so these transposase genes right here, or not transposase, avirulence genes, mind me, sorry. These avirulence genes were lost due to a transposon insertion, and avirulence genes are the genes that control interactions with the host. So if you mutate these or lose them, you can infect new hosts. It's like hiding from the host immune system. And then observation 2 is when Dr. Mark Farman, our primary investigator, observed that these transpositions were occurring in between 5S RNA genes. And then observation 3 is Gluck Thaler et al. Discovered giant Starship elements, novel DNA transposons, into 5S RNA genes. Which leads us to our study today.
All right, so now that we have a good background of what's going on, we are going to talk about our goals going into this study. So our first goal was to identify all the truncated and 5S RNAs. Our second goal is to record starships found between 1 to 75 and 56 to 116 5S RNAs. And our final goal was to categorize starships based on any similarities, if any at all. So 5S RNAs are elements that we find in the Magnaportha genome, and we notice that these 5S RNAs will split. So when they are intact, they go from base pair 1 to 116. And when they split, they'll go from 1 to 75 and then 56 to 116. And what's interesting about this split is you notice the 75 and the 56 coordinate overlap. So we get this direct repeat that is present on both ends. So, and going back, if we see that the starship is in between these, our first goal was to find where the 5S's are. That way we can predict where all the starships are in between. So the first method we used to identify all of the 5S genes, we wanted to identify all of the intact ones. We went to our Unix command line. Unix is a great tool. It's like Google per se. So Google, you type in what you're looking for and it pulls out results from a database. And this does the same thing, except we're making our own database. We're telling it what to pull certain aspects from. So here we're using a BLAST command. In BLAST, you will have a query. Our query is our 5S RNA. This is what we are looking for within the genome. Our genome here is U233. This is our subject. So this is what we are looking for, the 5S elements inside. Then we use an E value, 1E -10. This is very stringent. We want a smaller E value because that means any of the results, the alignments that we're picking up, are less likely to be from happenstance and more likely to be on purpose. Output format 6. This allows us to see what data we are interested in, the order of the data, the way that this information is organized on the screen. we use the command grep 5s. That just means all the alignments we see will have the title 5s in them. And we use the awk command. Now this was the most important because this allows us to see only the 5s alignments that we want to see. So here $7 is corresponding to column 7, and we want that to be the number 1. And we said we want column 7 to be 1 and column 8 to be 1/16. So here you can see column 8 has 1/16. So that way, when we actually run the command, all we see is all the 1 to 1/16s within the U233 genome. And this goes on forever because there are lots of 1 to 1/16s in the U233 genome. So now that we have our fully intact 5s's all written down, we want to find the ones that have split because those are the interesting ones and the ones that will lead us to our starships. So the command is the same at the top, other than what we're specifically searching for. So now we're using awk. We want column 8 to be somewhere between 70 and 80. So here I highlighted where it has done that. We have 75, 75, 75. And then we also want column 7 to be somewhere between 50 and 60. So now we have all of our 56s and a 51, which that one's an interesting guy. And I can talk about that a little bit later. And here the double pipes means or. So if I had the and symbol in there, then it would only give us alignments that go from 56 to 75. And that means we're probably not going to get a lot of hits because these things don't break up in that way. So by having the or statement, I get my 1 to 75s or my 56 to 1/16s. And we repeated this for every genome. And again, this is what we're looking at. So every time we found the 1 to 75, we have this half. And every time we found the 56 to 1/16, we're looking at that half on the far end. After doing this in every genome, 10, we found tons of the intact of 1 to 1/16s. 5' truncated corresponds to the 1 to 75s, and the 3' truncated corresponds to the 56 to 1/16s. And you can see those numbers tend to be pretty even in the results. Our second goal was to record the starships found between the 1 to 75 and 56 to 1/16 5SR RNAs. And just here's our reminder again. So now we've recorded all of these guys, and we're going to look for the starships in the middle. Now we're using a program called FACET. FACET is a very, very nice program made by our friend, Alex Stewart. Now this looks a little bit complicated, so I'll just show you the input here. We're running FACET. Our subject is U233, and our query is 5SR RNA. So it's similar to the blast that I had shown before. Once we run that, it sorts it all the hits by chromosome and outputs a GFF file, which is really important because then we can put that GFF file into a program called IGV. IGV is a genome browser, so we can actually go in, look at the nucleotides and see what's going on inside the DNA. If you look at the far end, you'll see we have a yellow and an orange bar. You might have to squint your eyes a little bit, but those represent the 56 to 1/16 and the 1 to 75, while all the blue bars represent the 1 to 1/16s. So if we zoom in on that far end, again, you can see here it says 56 to 1/16, 1 to 75. And in reference to the model I had shown before, we can assume that our starship is going to be sitting right in between those guys. Now, what's really important when we were going through these is that these 5S elements have to be facing in the same direction. So if one was facing to the left and one was facing to the right, we can't necessarily say that they were related. Maybe some transposition happened, an inversion, we're not really sure. So if they were facing in the same direction and within a reasonable length of each other, we could assume that they were once an intact 5S that got split by the starship. If we zoom in on the 5S to the left here, now you can really see why this is the genome browser, and we like it a lot. We can see all of the bases, and again, we will assume that the starship is everything after the 5S. After using IGV, throughout 10 genomes, we found 21 starships ranging from 10,000 to 350,000 base pairs. This is crazy. As Pat likes to mention, we expected to see, we'll say, a rabbit in our backyard. This is like seeing an elephant in your backyard. These things are ginormous. So our final goal, now that we have all of the starships, is to categorize them based on similarities, if any. So looking at the lineages, the host lineages these fall into, we would expect that if strains infect some similar hosts, they'll probably have similar starships. So in Ceteria, we have Arcadia and US71 that we predict would be similar. Oriza has FR13 and E11, and Lolium has FH and LPKY. And here you can see a host diagram of all the families grouped together. Okay, I need you all to, we're all going to take a deep breath because this is going to look a little wild. It's okay. I know it's crazy. This is MOV alignment. Surprisingly, it's probably one of my favorite tools that I use. This is all of the starships loaded into one big file. MOV can take on really big, large amounts of sequences. And so here is all of the starships compared to each other. Each line connects similar sequences. Can you not tell? Obviously. But you can see, you know, we have these big, the pink blocks that are the same. So in my best effort, while it looked like this, I tried to group all the ones that kind of had similar boxes together. Now I got this, still messy, but it's better. So after doing this, I was playing around with the tools a little bit more, found out you can just take away the lines and make the boxes solid. It's so much better. We're going to zoom in here on the far end. And now that we're zoomed in, we can see a lot more of the features here. It's a lot less crazy. We can identify potential deletion or insertion events because the starship doesn't have the purple box. The starship does. And then we also see we have these pink alignments again, they're a little zigzag. What's nice in MOV, you click on those, they'll line up for you real nice. Now, unfortunately, like I mentioned before, these things ranged in size from 10,000 base pairs to 350,000 base pairs. So comparing them on their similarities is really hard because some have a lot more content inside of them than others. So the way I went about it was I focused on their border sequences. What is the same on the very ends of the starships? And in MOV, that was really hard to do. So I moved on to a program called ClueStall. ClueStall takes on smaller sequences, but you can really dive into the base pairs. So again, here are all of the starships compared to each other. The stars at the top mean that they all share the same base at that position. And while investigating, we found inverted repeats present at both ends, which is very significant. All that means is that it has a line of base pairs that is flipped on the other side. It's the reverse complement. We also found linker sequences on the right end of the screen. You can see there were two present. Every starship had one, AAGAT or ATTT. We don't know the significance of these sequences at the moment, but there has to be some significance because every single one has them. And our final consensus of what most of the starships followed, they were between our 5S RNAs, like I mentioned before. They had inverted repeats on either side and a linker present in between the inverted repeat and the 3' 5S RNA. Our final characterization of starships, as I mentioned in our last goal, we figured starships that infect similar hosts would have similar starships. This turned out to be true for quite a few. FH and LPKY are in the group Lolium. E71 is in Trichticum, and those are very closely related. And they all had a similar starship, all on chromosome 3 from about 1.5 to 1.8 megabases. And Arcadia and US71, they shared two starships, one on chromosome 3 from 1.2 to 1.4 megabases, and one on chromosome 6 to 1.8 to 1.9 megabases. And after recording all starships, we assigned each starship a letter for easy reference. So here is our final characterization table. We grouped them by borders similarity. So each letter represents a starship. we found that most 5' truncated 5S RNAs are actually from 1 to 69 and not 1 to 75 like we originally thought. So in our original parameters using BLAST, we searched for 5S's that went from 70 to 80, thinking we would capture the 75. But after using IGV and really diving in base pair by base pair, we found that these actually go to 1 to 69, most of them. So we had to go back in and do some more research. Our 5' Starship end shows less conservation than the 3' end. So here we can see similarities between all 5 Starship categories. Not all of them have an inverted repeat on this end. Only one Starship has a linker on this end. While on the 3' end, they all have an inverted repeat and they all have some sort of linker. So we know that the 3' end is more conserved. The 3' inverted repeat loses a base when paired with the AAGAT linker. So here we can see the inverted repeat. You can tell it's missing a G here and it's missing a G here, and that's because of the AAGAT linker present. We also found 1 to 64 5' truncated 5S RNAs, and those were more associated with unique 5' Starship borders. So the only two that went from 1 to 64 on this side, theirs are completely different than all of the Starships that we found. So they got to be put into their own category. And we're still finding more of those currently. There's quite a few new Starships that we will soon be adding to the table, just because, like I said, our original parameters assumed that the truncated 5S would go from 1 to 75, and now we're finding 1 to 69s, and now 1 to 64s that we are still adding to the table, which is pretty cool. Lastly, new parameters, like I said, may reveal new Starships in new categories. A recent discovery that me and Ciara have found out yesterday is that we may actually have some 1 to 65s, which is really cool. So hopefully we'll get to talk to you all about that in a future seminar. All right.
I'm back. Aren't you excited? All right, so using sorry, no, you're good. Using Savanna's research as a guide, I decided to look at the gene contents of Starships. Now, when I began my gene content research, I was looking for a tyrosine recombinase that would be analogous to a transposase, as indicated by the second paper. Glut-Thaler indicated that they found Duff domains that were indicatory of a tyrosine recombinase. However, I did not find any of that, so our efforts moved forward to identify a transposase and look at the gene contents of these Starships because they are massive. Anyways, so our efforts thus far have focused on characterizing the gene content of B71, FH, and LPKY, which share the most synteny, and are also in the similar host specialized populations, with FH and LPKY being in lolium and B71 being in triticum, which are in sister things. I forgot the word. Anyways. All right, so how I went about this, I began with B71. So B71 was my consensus element. I went and got a better sequence that Dr. Farman told me to pick and extracted the already found Starship to get a better alignment and get a better read. I then took that sequence and put it into FGenish. FGenish is a program that predicts genes from a sequence. So as you can see here, we have B71. It was about 300,000 kilobases in length, and it had 35 genes predicted within it. And then FH and LPKY was helped to be predicted with the genes from the lovely Blake. So we worked together on finishing these up. They had 27 and 32 genes between them. So relatively close, like in number, to the gene content, but then we wanted to see which genes were conserved among these 3 strains, if any. So we then took the gene predictions and inputted them into predicted protein sequence here into BLAST-P and blasted them against a database of Magnaportho genes. So we put the organism Magnaportho right there and hit blast. And we then organize them by looking for a very low E-value, which indicates that it would be like less likely for this alignment just to be by chance, and then percent query cover and percent identity of the genes and the genes from the database. Some of the genes are associated with other transposons contained within the Starship. So as I mentioned before, the retrotransposons only their genes are very small, and they only contain like a GAG pol, an NV, or a GAG pol, or a reverse transcriptase. So as you can see here, this is the Starship, like this entire area here, that is our genome that we put into IGV. And these are the teeny tiny transposons. They look like little specks. So these are your typical transposons. And this is the big mama right there. So I went through and I documented the locations of all these transposons that are inside our Starships. And I documented the location and what genes were explained by them. So here's a bear your britches or whatever you said. Like, hold on to your britches. So I'm gonna blow them off. Anyways. LPKY, B71, and FH shared genes were put here. Blake made me a very nice Venn diagram in Adobe Illustrator. We have the pol GAG, type 1 PKS, cytochrome P450, that is for electron transport chains. And then we have some ABC, where is she? ABC, we have some secondary metabolite genes as well. So it's cargo carrying fungal genes that are secondary metabolites here, ABC transporter substrate binding protein. So we have some cargo genes being carried around the genome as well. So if I went through, and these are just based upon the genes we saw that had the same sharedness amongst each other. I went through and blasted all the genes against each other with a very stringent E-value and grep to see only 100% identity between the genes. And the ones that were 100% identical between the Starships were acetyl thymoserine, thiolase A, that's a big word. And this one and the next one shared 100% identity in all the Starships and the same location. So they were shared between all the Starships, and they shared the same location at the very beginning of the Starships. Then the BTBPOZ domain containing protein was also shared with 100% identity and length among all the Starships. The polykete synthase was shared with 100% identity among LPKY and FH. And the mitochondrial chaperone protein was shared with 100% identity between LPKY and FH. More was shared between LPKY and FH, as we suspected in the beginning, because they are in the same, or two strains of the same host specialized populations. So moving forward. So then I went about identifying the hitchhikers that explain some of the genes that were seen inside of the Starships. I began with B71, and this is kind of how I set it up. I found the hitchhikers, which are the other transposons. We're insulting them. So they're like riding along. Anyways, Retro 7-1 and Retro 2 were explaining the pol proteins in specific genomes, B71. MLGR is another transposon that explained a GAG protein in the genomes, and MGLR3 was also explaining a polymerase, and then the Retro 7, 1, and 2 explains pol proteins. So then I went about looking at the rest of the genomes and looked at FH, LPKY, and B71 and saw that they had some similar transposon activity, retrotransposon activity in here, and then MLGR or MGLR3 was shared amongst all three, but LPKY had a different transposon, Maggie, and B71 had a Retro 7 1/2 at the end. So mobility.
All right.
Oh, one thing is there was no transposase being found, but it may be one of the ripped up proteins because it's a repeat element.
Okay, so after we identified all the starships, one of the next questions we started to ask was about starship mobility. So we were wondering why these starships were particular to each genome and why they might have not been showing up on other genomes. We were wondering, was this due to insertion differences, chromosomal deletions? That could have encompassed why they weren't showing up. So what we did was, this diagram at the bottom is for what I will be referencing, explaining starship mobility. Here on the left, you have your 5' end of the 5S rRNA in the orange, and the yellow is the 3' end of that 5S rRNA. We refer to borders, as Savanna used to identify starships, as approximately 200 bases into the starship to identify similarity. And then the flanking sequences is the inverse. So it's 200 bases outside of the starship. Okay, so thanks to our friend Alex, we were able to use FACET. So in order to search this, I use a FACET command. First, I have listed US71 as the subject, which is the genome that we're looking in on our genome browser IGV. And then we're looking at the B71 chromosome 3 flanks for that starship. So what I did was I took the FASTA files for both US71 genome and those flanks, and I put it into a GFF file and put it back into IGV. So I loaded the US71 5S rRNA sequences along with those flanking sequences, and I found on the same chromosome, actually just kidding, I found on chromosome 2, so this was a different chromosome from where the starship originally was on B71. This is what we refer to when you zoom in as an intact 5S rRNA. So this is basically just those flanking sequences with the full 5S rRNA ranging from 1 to 116. So that means that starship has either inserted in it and left, or it was never there in the first place. All right, this is another example, which I'm going to explain later, but I use the same command, this time looking in the FR13 genome, looking at the G11 chromosome 1 starship links. This one is really interesting because you look and it's on the same chromosome, it's still on chromosome 1 of FR13, but this time you have the 5' 5S rRNA along with the flanking sequences for the G11 chromosome starship, but the 3' end and the flanking sequences on that 3' end are deleted. So that's something we'll look into later. All right, so with a total of 21 starships and 10 genomes, we did 210 comparisons looking at this. First off, I want to point out all the white sites, which is what I mentioned earlier, being the intact 5S rRNA. So it was the full 1 to 116 5S rRNA. This showed up exactly 100 times. So that equates to 47% of the time. So that was pretty common. A few other things that we saw were just a slight gap between the 5S rRNA and the flanking sequences, or we saw the chromosome translocations, like that first example I showed you. We saw a few borders being deleted, like that second example I showed you. And we also identified orthologous starship insertions, meaning that there might be potentially new starships with similar lineage. And then we also identified some special cases as well, which were a little bit different than that. Okay, in addition, we identified starship trends. So this was where a certain situation was the same for every single starship. This was seen on FR13 chromosome 1, U233 chromosome 4 for both of those starships, US71 chromosome 1 and Arcadia chromosome 4, which we're going to look at right now. So for Arcadia chromosome 4, those flanking sequences compared to every single genome showed up on B71, but on chromosome 7, the 3' end was flipped along with the flanking sequences. So what you're looking at with this diagram, the middle line right here is just what it looked like in IGV. I just made a cartoon from Adobe Illustrator to put in here, but that's pretty much what it looked like. If you were just looking at our genome browser, at the top, this is B71 chromosome 4, which is where I first went to look, assuming that the original starship was on chromosome 4 of Arcadia. It matched up to that 5' end, but then looking on chromosome 7, we found the flip 3' end, which is a trend. So it was on every single genome. All right, this is that second example that I showed you all earlier about the G11 chromosome 1 starship on the FR13 genome, where that 3' end along with the flanking sequence was deleted. Again, this is kind of a cartoon version I made from what I saw on IGV. I'm going to dive into a little bit more of what we saw, including this internal deletion. So actually FR13 contained approximately 112 kilobases of sequence that wasn't in the G11 Starship. So maybe that suggests that it was on FR13. And then these sequences right here, the blue and like the yellow greenish colored recombined on G11. So I thought that was pretty interesting. I just kind of stumbled up on that on accident, but that's really cool. And then this possibly is what we think explained the 3' end deletion. This is an intermediate figure. So there's an MGL repeat sequence on G11 sitting right outside the Starship, as well as a sequence right on the end of the Starship. So we're thinking that since these sequences are similar, or if not the same, on FR13, they recombine together, as you can see here, eating out that 3' end of the Starship. And that's the picture once again. So those are just a few examples of what we were looking at when it comes to Starship mobility.
So that basically concludes the current research for our group. And our future directions do include continuing to analyze and compare the Starship data, as well as modeling the transposition and then mapping out genes for the Starships as well. The reason that we do these seminars is so that we can have input and questions on our research, because that fuels future ideas and future questions for our research. So thank you everyone for being here.
Thank you.
Yes, of course.
Yeah.
Before I ask the question, just to clarify, non-aligned full line sequences with similar characteristics, like whatever you would put, right? So what is the benefit of putting everything into facet first to identify like predictions before putting it into model line? Is it like just the size of the sequence?
So facet is what I use to just identify where they are, more or less. And mob is what I use once I found where they are. I can take all of those sequences out of IGD and put them all into mob, and then mob will say, okay, so you've identified all these. I'm going to tell you where all of these are similar internally. Is that?
Yeah.
I've got sort of a more just broad question, make sure I'm understanding this. Transposons are parts of a genome that move from one place to another.
In mitotic division, yes, they move from one place to another. So they're not just like floating around the genome, but in mitotic division, they move around the genome. And this thought they came from retroviruses. So that's their ancestral origin, and then they lost their capsid. So therefore, they're just like now genetic consistency.
And so starships are just really big ones.
Yes, they are massive.
And you're interested in those because you think they could explain why this asexual organism infect different hosts.
So we think that, yeah, it also causes, it's a known thing that transposons are used by pathogens to induce variability among their offspring. And then these transposons are also cargo carrying, so they're carrying secondary metabolites, which could induce them to be mutated at a higher rate by putting them in a different chromosomal location that's less bound up by gluten. And then another thing is that starships or transposons in general just induce mutations in the DNA, like inversions or cause like non-homologous in repair or stuff like that.
So the end goal maybe would be to figure out what in these transposons is allowing them to infect these crops and therefore try to combat that.
Yeah, so you can see that in the first paper identified that 2 avirulence genes were absent in the triticum strain. It was like PWT6 and some other avirulence gene, there's many of them, that were absent that allowed, or it's been hypothesized to allow that strain to affect wheat. So that was a host jump. So it has been demonstrated already previously in that paper that they have deleted avirulence genes, thus allowing for host expansion. So you can already see the adaptive benefit of the wheat.
Yeah, I just wanted to, thank you.
Yeah.
So Alex, this gets to me with around. Hermomules, it is special organisms since they can't undergo recombination in meiosis, but eventually decay and go extinct because they are limitations. The yellow rock birds are asexual. There's no males found in the rock birds. They do very well. It's felt that transposons may help overcome the lack of sex in rock birds and same here then. So these transposons may allow systems that are mostly asexual generate variability to intergenomic recombination, moving genes back and forth.
Which they can be acted on by natural selection as opposed to recombination. Okay, that makes sense. Yeah.
They have many offspring, so to sacrifice a few would not be a big deal to them.
Other questions? Or okay, next slide, please. So these guys had a brilliant idea of giving a seminar in the UK, which they did. Okay, and they did in a similar Department of Nanotechnology in the UK a couple weeks ago, and we got some feedback. Mark, say you guys, EKU Pride, that's what we do. Okay, and we're good. Okay, so my colleague Mark, I sent him this text message. He was in England at the time. He zoomed in for us, and he got rocked. And then below that, hi Mark and Pat, that's from Dr. Costu. He is internationally known plant pathologist, where he works on plant defense mechanisms. He basically said these undergrads and our one grad students were a great model for their grad students that the kid I had to do research. So bottom line, folks, be proud of Eastern. These guys rock UK, and actually at several inquiries about can your students come here for graduate school, you want to get your people out here because they can really stir the pot and bring in a great perspective on your research. So UK thinks we're pretty good, folks, and I think that's a source of pride for all of us. So on that note, I think we have to end our seminar. Thank you all for coming. Hi, grads. My team has some class after this. We have to go to the case for lunch, go right ahead. It's gonna be quite a lot of skills.